Synthetic biology is almost a natural continuation of what was called metabolic engineering before, or just biotechnology, and the boundaries are extremely gray. These are all very overlapping terms, and the goals are often the same. At least when it comes to application, we want to change a microorganism's behavior towards something that we feel is beneficial for our society. The strength of synthetic biology is its ability to look very focused on a small aspect, on one circuit, and to understand that circuit in detail. And if that was to work, then the ideal, or the principal idea is one could take these circuits, put several of them together, and eventually get something larger, a larger machine to work. A little bit like an engineer would work, or maybe exactly like an engineer would work. The problem is, this does not really work so well because we don't understand our machine, our microbes, as well as an engineer does understand their machines, and the circuits, to just put them together, don't really work currently so well. At least not when it comes to application. So that's really what the question is at the end, and the challenge has been the same for all of these areas, from biotech over metabolic engineering to synthetic biology. You need to make more of something, and you need to make it at a commercially competitive level. And that turns out to be extremely difficult, and it hasn't really, in my view, not changed dramatically over the course of the last 20 years. So the big question is, what is missing? What do we need to do to be more successful? And that's not so easy to say. What I think is clear is the fundamental problem is we are not understanding our machines, the machines that we want to engineer, our microbes. But we don't need to, for a biotech or synthetic biology purpose, we do not have to understand the whole organism all the time. But what we need to do is we need to know what are the key elements, the key regulators, and what precisely do they do. And our understanding has to be quantitative. We have to be able to make predictions, dynamic predictions, like if you change the level of a regulator or a binding site of a regulator, how is that going to affect the protein levels in the cell? And that's a challenge, and I think it will require, besides very good knowledge and understanding and good methods and experimental techniques, there's no way around having good computational models, predictive computational models, so that we can learn from not being good in predicting.